Oliver Smith has won his Nobel Prize for discovering the process of gene targeting. And now we can use that technology to ask what is the function of a gene in the disease or health of a human being. I understand that Oliver Smith is well into his 80s. He's a very great example of how life should be lived. You know, the excitement should be kept throughout your life, and there's no reason to stop just because you achieve the greatest thing that a scientist can achieve.
I'm going to share with you the experiences of a graduate student who has never grown up. Because I still work at the bench, and I'm not the chairman of anything, and I don't direct any laboratories, so I am still a child of science.
So the research that I do in a broad sense is I'm interested on understanding the mechanisms of appetite. More specifically, I'm focused on a cell type in the lining of our intestines that secrete hormones, and these hormones regulate appetite by talking with neurons in our guts and also neurons in our brains. This question is at the core of understanding how we can solve problems like obesity.
Good.
So I'll tell you a little bit about me.
Yes, do. Yes, yeah. I want to hear.
So I'm from Ecuador.
Yeah.
I grew up in a very small village in the jungle of Ecuador.
Oh, very good.
Yeah. So then I decided for college, I decided to study agriculture. That's when I became interested about doing a little bit of research. So there was
once it gets in the blood, you know.
Yeah. Then you have to keep going.
You can't get rid of it.
Yeah. So in fact, I started working with turkeys.
Oh, turkeys.
But then eventually I got interested in nutrition.
Yeah.
My professor, he came up with the idea that you know, it's very important to feed as early as possible the little birds, because once they hatch, they are not able to find the food and then they die.
Yeah.
So he came with the idea, why we don't feed them while they're in the egg? So we can inject some nutrients into the amniotic fluid.
Yeah.
And then, you know, maybe see what happens.
Yeah.
So he did that, and then he found some interesting things.
What did you do? I'm not interested in what he did. I'm interested in what you did.
So I wanted to apply that to see how that affects the development of the digestive
So did
tract.
you do some of that work?
Yes. I became interested about appetite like 3 years ago. As I was doing an experiment in my PhD, I was feeding embryos, putting some nutrients in the amniotic fluid, and one of the things that I saw is that when they hatch, they were hungrier, more active than the controls. So because of that, I started to read about appetite, and particularly how is that the intestine is able to recognize nutrients and tell the brain, well, we got enough food, stop opening the mouth. This hormone is produced by these enteroendocrine cells, along with another hormone, glucagon-like peptide 1, that stimulates insulin. So both are very important in obesity and diabetes. So what I start to do is just take pictures of them using a confocal fluorescence microscope. These cells were thought to be just triangular before. The first thing that I start to notice is that they had a little arm at the base. The arm was actually really long, ended in a bolt-like, looked like an axon, and was going underneath. So then I thought, well, these must be connected to neurons. Mice.
Well, you know, there's a network of neurons in the intestinal wall.
Yes. It's a second brain.
Yeah.
So the part that I'm really interested is how that these cells communicate these signals to the neurons that are laying on the intestine.
You know about the W locus and whether that set of receptors and ligands, you might say, controls the development of that neural network in the gut. It's been known for quite a time. You should go and read about that because you ought to know. Yeah. And animals that have that mutations in that gene, their gut doesn't have the normal peristaltic waves because it doesn't have cells of the same sort that you're describing. I'll bet your cells are part of that network. Wow. So you should learn about that.
So this part that Oliver Smith has just shared about how a part of the genome drives and guides mother cells for them to become neurons and lay on the gut wall. It's really important for my research, although it's not directly related to my research, but it's really important in terms of understanding how this one player is born, raised, and once it becomes an adult, interacts with the other player, which are my cells on the gut wall that secrete hormones. I thought that they must be connected to neurons, so I did the immunohistochemistry. Mm-hmm. And doing the reconstruction, I was able to find that they are actually connected at the base.
With the synapse.
I think that is a synapse.
Yeah.
But now I'm in the process of proving it through electron microscopy, trying to fish out that synapse.
Well, so I like the story, but I do think you're going to find that it interacts with the system I'm telling you about.
What are you working on right now?
Well, I'm doing some crazies. You know, when you get older, you can do crazy things. I'm actually making tiny gold particles, nanoparticles, and using them to study how the kidney separates big molecules from small molecules. I have a good postdoc who's helping me with that. He does the animal experiments and the electron microscopy, and I make these gold particles. I've just learned how to make them different sizes, and so that you can get, make some that are small, like small proteins, and some that are about the size of albumin, and some about the size of immunoglobulins. So I can make them different sizes. And then you can actually see them in the electron microscope if you use high enough magnification. So you're really seeing individual molecules. It's it's exciting.
It's really exciting.
Yeah, and enjoyable to do. I haven't got anything published yet. It's terrible. We've been working on it forever and we haven't published a thing. We're going to try this summer and get a paper out, but it always seemed like there's another question to solve before we're ready, you know.
There is always something
always
to
another
follow
question.
up.
Yeah. And there's a big debate as to where the separation is of proteins from the other fluid, like salt and glucose and water and so on. Because the kidney doesn't excrete albumin or antibody. You don't lose your antibodies every day. And somewhere there's selection. And there's a big debate on where it is. And I have one idea and most people have a different idea. So I'm working uphill. At the moment I have a range of about 12 or 15 tubes that I've made, each with a different size particle, ranging from small to big. And I tell them, take one of the small ones and the biggest and do that. And then we'll work our way in with it. Because I think the real one is in the middle. And by then we'll be experienced and we'll know what we're getting. So anyway, that's what I'm doing.
That's really exciting. It's kind of like how they found the size of the tight junctions in the intestine.
Yeah.
You know,
using different molecules.
Different molecules. Like how is that the glucose can pass, but there are others that cannot pass.
Right.
You know.
I'm an inventor. That's the answer. I invent things, and I like to make things and do things. So experiments that I do myself are always more enjoyable than experiments that other people do, which is rather selfish, I suppose, and self-centered. But it's really not.
I want to ask you about that. You mentioned that at the very beginning, your equipment was very, very simple. Do you think that had an influence on your drive for creating new things?
Oh, no. Well, I don't know whether it influenced me. I never thought of it that way around. I think it was usually the other way around. You needed to do something, and so you made something to do it. You know, in Toronto, I needed electrophoresis. I remembered helping my mother do the laundry when I was a child. And when she cooked this, she cooked starch up to starch my father's collars, and it made a gooey liquid. And when you tied it up at the end of the day, it was a jelly. And I thought, if I cook the starch and make a jelly out of it, then I can let the protein migrate through the jelly and stain instead of doing 40 protein determination. So out of laziness, I invented gel electrophoresis.
And so most of my apparatus has been very simple.
But the ideas are big.
Well, I don't know about that. They work. I mean, that's the thing, yeah, so.
What do you think that are going to be the new genetic tools that are going to allow to ask these questions, like maybe one gene at a time, a path of genes?
Oh, you're asking a question I don't know the answer to. I mean, if I knew the answer to it, I'd be doing it. So you're going to be the person who finds the answer to that question. It's your turn.
Now
I've done my turn, now it's your job.
That was something they used to label equipment with when I was a graduate student. If there was a piece of junk, it would have this label on it. No bloody good. But okay for Oliver. Leave it around and I'll use it.
Another thing that I want to ask you was about collaboration. At some point I listened to your lecture, your novel lecture, and I know that collaboration was a big part of the process.
I was talking at a meeting in New York. I said, I have this collaborator, Francois Allongeur. I said, I've never met him. And a guy in the back of the room gets up and says, me. so I met my collaborator Alain Gillas that way. So this is what collaboration is. It's really, it becomes a joy. And there's a big secret to it, which not everybody knows. And the secret is when you are working, if I'm working with you and you are my collaborator, what I have to do is always think about what is in this collaboration for you, not for me. If I think about what's in the collaboration for my collaborator, it will work. If I think about what's in this collaboration for me, it won't work, because I won't be looking after my collaborator. And then if they do that in a collaboration, it's bound to work, because it's advantageous for everybody.
That's an advice that I'm going to keep it in my pocket, because I'm, as a postdoc, that's when you start to send emails to people.
You need help, yeah. And never
help,
hesitate to ask help. Most times you'll get it.
Is there a possibility that I can visit your lab? That I can
of course.
come and visit? And I know that our fields are not exactly the same thing, but I'm using your tools.
You just come and visit sometime, and you probably find me at the bench.
So I will definitely take advantage of his invitation, but I think that I want to think about it for a couple of weeks. Most of the things that I'm interested on is getting some advice, not specifically on the science per se, but how to deal with science in general.
The best weekend is, for example, on Saturday. I'll fly my airplane in the morning, I'll take my wife to lunch in the middle of the day, and I'll do an experiment in the afternoon. And that's what I consider a good Saturday. I can give you a little piece of advice. Remember that whatever you did as a graduate student, whatever you did as a postdoc, you do not have to do that for the rest of your life. You don't have to do it at all. That's training. What you do now is what's in here. Not what you learned, it's how you learn to use what you learned to do something new. So you can go into something quite different. You don't have to stay doing the same sort of thing.
I just spent a whole hour with a Nobel laureate. It was a tremendous experience for me as a young scientist.
It's very important to find something that you enjoy doing, because a lot of the time it won't work. And your chance of making a great discovery are quite small. So if you depend for your enjoyment of your science on making some great discovery, you're going to be unhappy. because most of the time you won't make a great discovery. You might never make a great discovery. It doesn't matter. If you're doing what you enjoy, you can have a grand time and you can make a contribution.
When I go back to my lab, I will keep doing what I have been doing and what it brought me here, the excitement about science. I'm really excited to be at the edge of knowledge, be able to discover and advance a little millimeter and know something new. And that's what I'm gonna keep doing, and that's why I came here.